slide going. Let's move to the next slide. I'll start with Leon Tolstoy. That has nothing to do with Exposome, but some of the things he wrote. I was reading War and Peace about 3 summers ago, and at some point in time, towards the end, when he's philosophizing a lot, he says, man's mind cannot grasp the causes of events in their completeness, but the desire to find those causes is implanted in man's soul. And without considering the multiplicity and complexity of the conditions, any one of which taken separately may seem to be the cause, he snatches at the first approximation to a cause that seems to him intelligible and says, this is the cause. And he was talking about why would a French soldier march, through the mud across Europe to go and die in the Russian steppes and the Russian front. But basically, this is really appropriate to some of the things we're doing, especially in exposome research, and where we are looking at complex relationships, and we have to be aware that biology and society is complex, and we tend many times to just do what Tolstoy is saying, saying this is the cause, when really causes are much more complex. So we have to have this in mind. And we have this in mind, and there are methods for evaluating complexity, and we use them much more now than before. So let's move forward. Next one. So in the 90s, we had the evolution of what was called molecular epi, and it took care of the biomedical part of complexity. So we had new techniques, laboratory techniques, and we could use them in large epidemiological studies little by little. Before we could do just few analyses, little by little we could do many analyses. So we developed biomarkers and genetic analyses. And then at some point in time in the early 2000s, and I will come back to the to Chris Weil's definition of exposome. Chris Weil was the one who proposed the term. We expanded in a way molecular epi, not to be only the part of what happens in the cell or in the molecule, but also exposome would cover everything from complex exposures to complex biology. And so what molecular epi and exposome do is they promote the incorporation of advanced methods for exposure assessment, molecular, genomic, and biochemical concepts. And this is done in well-designed epi studies, epidemiological studies. That's what defines the exposome. Next one. So this is the classical approach that we have been using all our lives in epidemiology. So we have some kind of environmental exposure assessment. Environmental, I mean non-genetic. It can be lifestyle, it can be anything. And then we have some kind of clinical disease, and we make an association. And using this model, we have made enormous progress, and we have saved, millions and millions and millions of premature deaths. The next one, as techniques developed, we obviously could fit in what happens in between the external exposure and the disease, and measure internal dose, measure early effects, measure susceptibility to exposure, to developing effects, to developing disease. And so we have the capacity of looking at this much more complex relationships. The next one. This is one of the papers I think I put in the Moodle. It's an old paper we wrote with Neil and some other colleagues a long time ago. And we were skeptical at that time about some of the use of the biomarkers. So this article, it's wrong in some aspects because it's too critical, but it's right in bringing up basic epi concepts that should be used in these studies. The story of how we measure past exposures, how we measure temporal variation, study size, confounding, and I will refer to some of the concepts we were referring to in this paper. Next one. My conversion, if you want, to biomarkers happened in 1994. When I was participating in one of these IARC monographs. These are the meetings where of the WHO meetings by the International Age for Research on Cancer that evaluate carcinogens. These are the official evaluations. And you evaluate all the evidence, the human, the animal, the mechanistic data, the exposure, and you try to make sense out of the whole evidence. And so at that meeting, we evaluated the epidemiological studies, and we thought they were not entirely convincing. And ethylene oxide is important, and it was even more important in the past. We had them in our hospitals where we were using ethylene oxide to sterilize. And so there were a lot of medical personnel exposed, and it's used in industry. So the epidemiological studies were not entirely convincing to tell us, yeah, it is clearly associated with, in that case, lymphomas. So the evaluation was limited evidence, not sufficient evidence, which is the highest sort of type of evidence. For animals, it was clear. And then we had the mechanistic data, the mechanism, and it was a time when molecular epidemiology was growing, and we had for the first time pretty good studies of exposed workers. So it is not something that has been done only in vitro, or in cells, or whatever. It was measured in exposed people, and they could show that exposure to ethylene oxide was producing effects. They could show that there were adducts, or there were some kind of gross chromosomal effects, and this evidence was used for the first time by IARC to combine with the epidemiological studies, the animal studies, and say, okay, the epidemiological studies are not entirely convincing, but we have convincing animal data and we have convincing mechanistic data. So we do an overall evaluation. We are sure that ethylene oxide is a human carcinogen. In the past, this would not have happened. And this is why these type of studies on molecular epidemiology and the exposome are important. They are important because they help us evaluate better causality, and because they also affect policy, which is effectively what we want. So that was the time when I said, okay, if I do these studies, I will be more capable perhaps of convincing people that something is hazardous, and we will take, it will help us take prevention measures. The next one. Next slide. So problem with historical exposures, going back to the article that Neil and some of us wrote at that time. Many of the biomarkers we are using it can be cholesterol, it can be lead, it can be whatever. Many of them indicate relatively recent exposures. So the problem is that if you do them in a case control study and you want to find out something, you have a chronic disease, you want to find out what happened many years ago, this doesn't help. But we have nested case control studies in cohort studies. So, and we have stored samples. So this solves the problem. That's why we're doing a lot of biomarker analysis in nested case control studies, because we have samples from the past. Obviously, again, if you have a lot of variation in the biomarkers, then getting just one measurement may not be enough. If you do one measurement 20 years ago, you have to make the assumption that that's a pretty good average estimation of your exposure. If there are things that vary during the day, it would change a lot whether you measure it Friday afternoon or Monday morning or whenever. So you have to do a number of assumptions, but we have knowledge and we can evaluate these assumptions. Next one. Here you can see the half-life of selected chemicals, and we're measuring lots of chemicals in occupational studies. So half-life is the period it takes for the amount of a substance undergoing decay to decrease by half. So you get a high exposure, you go into a room where there are people smoking, you get exposure to secondhand smoke, you inhale a number of compounds. Half-life for each of these compounds is the time that your levels will go down by 50%, grossly speaking. And you can see from the x-axis that chemicals have really very, very different durations of half-life. There are some that are metabolized in minutes and some that are metabolized in years. So if I'm doing a study on styrene, for example, to look at effects in the eye or whatever, and I get a urine sample a Monday morning, that won't be very good, because if the person hasn't worked on the weekends, their exposure from Friday has disappeared practically. If I do a study on, say, cadmium and I get a blood sample, I can go many, many, many years after exposure and I will still get an evaluation, a pretty good evaluation of chronic exposure. Let's see the next slide, which is the next one, which is an example from dioxins. Dioxins are and furans. This is the whole compound of TCDD and a number of very, very toxic chemicals. And the most toxic of the dioxins, TCD, has a half-life of around 7 years. So we are exposed through the food normally, the general population. This is the green part of the graph. So little by little we go up, but we don't get very, very high exposures. A worker, say in a pesticides plant or somewhere where there is an accident, a chemical plant, they may start working, their exposure goes quickly up because they have higher levels than the background, then they stop their exposure and it doesn't drops immediately because the half-life is 7 years. So it drops little by little. So if you think of it, the people, the workers are exposed in cumulatively in what is in this area under the curve. And this helped us do the big epidemiological retrospective cohort studies, the international ones from IARC, and they were done in many places in Europe, in North America, in the US, in New Zealand, in Australia. And we did the studies, we got blood samples many years after their exposure had finished. But then, since we had complete work histories, we could reconstruct their work histories and we could make an estimate, and we had the histories of the plants, the chemical plants and the pesticide applications. So we could reconstruct their history and we could do with modeling, we could reconstruct the curve for each of the workers by measuring their dioxin levels many years after the first exposure. But this we could do because the half-life is 7 years. And these type of studies we did, and eventually dioxins were classified as human carcinogens, and we're taking very, very strict measures to control exposure, also because of these studies. The next one. Let's see another example of a biomarker. This, I can be critical of this study because I'm a co-author and because Jordi Sunyer is a good friend of mine just next door. So this is a study on cotinine. That's what we measure in urine, as a substitute, as a measure of exposure to tobacco smoke. So urine cotinine levels in mothers at the 32nd week of pregnancy and birth weight in the birth cohort in Spain. So we measured cotinine at that week and we associated with birth weight, and the reference is women with practically no exposure, less than 4 nanograms per milliliter, and that's the weight of their babies, 3,384 grams. The low exposure, 4 to 20 nanograms, basically the same as the reference group, 20 to 50 nanograms, 20 grams lower, more than 50 nanograms per ml, less than 143 grams on average. So that's a pretty big reduction in birth weight. Does anybody know half-life of cotinine? Okay, it is less than a few days, actually, Emily, and it is very, very, very short. So if you think of it, we did one measurement and say it is 1 to 2 days. It's basically of that order. It is less than 2 days. Is this useful, what we did? We did just one measurement during the 9 months, and this is a chemical that has a half-life of, say, 1 day. do we get something which is meaningful? And why on earth do we get a correct answer? Correct, a posteriori, we know the answer in reality. So is it advisable to, if you want to look at this, to do it as we did? Exactly. Ryan says the correct answer. For regular smokers, they would continue to have high levels. These are non-smokers, all of them, but they are in the company of regular smokers. So in many ways, this is not a good example, because cotinine, we would have to have repeated measurements during the pregnancy to get an average of their exposure, not just one. But what happens is that you are exposed basically to sort of a standard level throughout your pregnancy, at least throughout the first part of your pregnancy. People get more sensitive at the end of the pregnancy. So what we got is one measurement, but in fact, it captured quite well what they were exposed, because they were continuously with regular smokers. They were not smoking, but they were with regular smokers. So this is not a good example. We got the right answer, but we could have got the wrong answer if this was not the case. So sometimes it works. Let's move forward. I'll skip this one because we are going late. So let's think about, what does a biomarker measure? You measure it. So you think it's okay because you measure it rather than you use a questionnaire, which is okay as a thought, but not necessarily correct. It depends what you measure with a biomarker. The fact that you have a biomarker is not by definition better than not having a biomarker and using a questionnaire or having another biomarker. So in essence, many times you want to control the external exposure. So you would like to have some kind of correspondence between your biomarker and your external exposure. We don't always have it well, but if we want to control exposure and take prevention measures, then we would like to correlate our biomarker with our external exposure, have an idea, what does it mean? So, because that's what we will eventually control, the external exposure. So that's what we would like to have. Next one. So in a biomarker could be in the causal pathway. This would be perfect. So you have exposure, you have a biomarker, and then you have disease. You measure the biomarker, it is correlated with disease, correlated with exposure. This is, a great biomarker. Next one. Sometimes it may not be the causal pathway. This is the example I skipped, actually, but it is correlated with something that is in the pathway. The example was on a type of adducts, hemoglobin adducts, which is just a connection of a chemical with hemoglobin. It's a covalent binding of a chemical with hemoglobin. You're not really interested in hemoglobin for some of the things, but if hemoglobin is M2, and this gives you a very good idea of what happens in M1, that could be DNA, then that's good enough. So we can measure something which is not in the causal pathway, but is strongly, very closely correlated. So that's good enough. Next one. But sometimes we can measure a biomarker that is really irrelevant for our causal pathway. It can be correlated with your exposure. That's also important. But it doesn't tell us much about what happens between exposure and disease. So it just tells us what happens with exposure, which may be the objective of your study. It's a biomarker of exposure. It doesn't tell you what happens between exposure and disease. The next one. And sometimes, and that's an issue, it could be a side effect of disease. If you do a study, a cross-sectional study or a case control study, and you don't have samples from a long time ago, then what you measure can be something which is related to, which is produced by disease or affected by disease. If you think of it, it's the same concern we have with cross-sectional studies with the questionnaires. You ask about sleep and effects on disease. Say you do a study on night shift work, and you don't know whether the people, is it first disruption of sleep that produced the disease, or is it You have the disease, which affects your stress levels, which affects your sleep. We can find it out, but it is essentially the same issue. So if you don't have prospective samples, you have to be aware that what you measure may be affected by the disease, or it could be confounded by some other exposure. So let's move forward. Next one. Study size. Study size is an issue for many biomarker studies. It was much more an issue before. It is not an issue for genetic studies because we have huge genetic studies. Next one. For example, this is just one of the many a study on height. What defines height genetically, and you have nearly 200,000 individuals, and we have lots of that. Next one, where we have this is a study, the classical study on asthma, the Gabriel study that lots of us participated, and you have studies with many thousands of participants, and you need very big studies because you're doing a lot of comparisons. In this slide, you have about 500,000 p-values. So if you're doing 500,000 p-values, you better have a big sample if you want to get results that are not entirely random. And that's what you see, the significant level, the nominal significant level is put to something like 10 to the minus 8. So you do some kind of corrections, which is classical in many of these studies on omics, where you do thousands and thousands and thousands of comparisons. Next one, and the next one. So in one of the articles I think I've put in the Moodle is this very short article by David Hunter, which is very nice because it talks about key characteristics of GWAS studies, genetic genome-wide association studies, which is studies that are agnostic. You don't have a prior hypothesis, you just do lots of comparisons and then you try to make sense out of it. And there are some key characteristics, large samples, and that's what we're going. And this is what we need for many of our studies on the exposome. We have new methods of collaboration, which is now entirely established. This was not the case 20 years or 25 years ago. We were keeping our data. We were not making them public. We were not collaborating as much. We were collaborating, but things have changed a lot, and it's really open to collaboration. And to a large extent, this came, genetic epi helped a lot to that. And for this type of studies on genetics, relaxation of methodological orthodoxy, genes, you're born with them, you die with them. There's no big confounding. Well, there's confounding by race or by a few other things that you can control, but you don't have all the other issues that we have on studies in epidemiology. So you can do pretty crappy methodologically studies. Sorry for using this word, but it's like that. Pretty bad studies methodologically. You can do these studies without knowing much epidemiology. You know genetics, you don't know much epidemiology, and they're okay because genes are stable. As soon as you do gene-environment interactions, you're lost if you don't know epidemiology, because there comes all the story of selection biases, information biases, confounding. So as soon as you get out of pure genetics, and that's where all the studies on exposome are outside pure genetics, then you really have to know epidemiology. The next one, please. And the next one. So these studies, with these studies, we have identified very small effects because many of the genes have a tiny effect. And also, if you think of it, for many of the studies on environmental, more than in occupational, we also have pretty low risk estimates. And we need to get very big studies to find them. For air pollution, for general mortality, the estimates we get is of the order of 1.06, 1.05 per so many micrograms per cubic meter of particles. So, and to find this, you have to get big studies. And it doesn't mean that they are not important, because if everybody is exposed, then the attributable risk is huge, even though the relative risk is low. So what we do in genetic epi, we also have to do in occupational epi and environmental epi many times, because we're interested not only on the very high risk, but also in the low risks, relative risks. Next one. Next one. Let's move to the exposome. So this is a sort of my pictorial description of the exposome of what Chris Wilde described. Chris Wilde was the director of IARC at that time. And he eventually said, we are putting, and he comes from the molecular side, from the biomarker side, but he knows epidemiology. So, and he was saying, we are doing a lot of effort on the G, on the genomics, and we are not doing enough effort on E, on exposures. And unless we understand exposures, we will never be able to prevent efficiently disease. So, and he was talking about recognizing the disparity in current knowledge between, he defined the exposome in a very ambitious way, all environmental exposures, including those from diet, lifestyle, and endogenous sources from conception onwards. So let's see the characteristics of the exposome studies, which is a key. If you think of it, exposome is epidemiology in the 21st century in some ways. You know, it's a simplification, but more or less that's it. Next slide. So the futures of the exposome, next one. It is holistic. We want to evaluate many different types of exposures. We want to evaluate the general external exposure, social class. We want to evaluate specific external pHs, benzene, and we want to also evaluate the internal exposures, the metabolic futures, microbiome, etc. The next one. Next. It is dynamic. We have understood that in many of our studies, we are evaluating just what happened the last few years. But then we know that for many of the chronic diseases are defined by what happened a long time ago, not what happened only the last 10 or 20 years. And we have a lot of evidence that early exposures affect late disease. So we have to move also for many of the diseases we're studying to a life course approach, if we can. It's not easy. We don't have, it's difficult to do prospective studies starting with early life, if you're evaluating prostate cancer that occurs, after age 65 mostly. Although we have studies that are based on registers that do it. Next one. We are using in exposome studies new technologies. New tools and technologies are a key aspect of exposome. We have all these new methods, and if you use them intelligently, in an intelligent way, you can do much more powerful studies. We don't use new tools and technology just to use them. That's not our purpose. We use new tools and technology to make better studies. But there are lots of them, and if you're not aware of what's happening, you just lose capacity. Next one. We try to integrate biological responses, the internal exposome, with the external exposome to do the continuum. And exposome studies use a lot untargeted discovery. You just don't go one exposure, one biomarker, or one disease. You want to evaluate to have a wider view, and you can go untargeted. Evaluate lots of different exposures, lots of different biomarkers, many diseases. Next one, please. This is an example from the early life exposome study, Helix, where they had, it's an early example, actually, we have many more exposures now. This is from the very first analysis, where you would have in a sub-cohort from this big study of 30,000 people from 6 countries, 1,200 children, and at that time we had 90 plus exposures. Now there are many more, and you can see the different types. There are water, meteorological, tobacco, there's occupation, there is phthalates, built environment, organophosphates, whatever, air pollution, and you can see their intercorrelation. Much of it is because of confounding by country, to say the truth. So you adjust for country, and lots of these associations disappear. But it's obvious that exposures are intercorrelated in the occupational and in the wider environment. So, and if you just measure one exposure, you have unmeasured confounding. If you measure lots, you are more capable to control for other type of exposures. In the occupational environment, it is true that you have less of these interconnections because you have high exposures to few agents, and it's easier to measure. If you move to the general populations, you have many more of this interconnection between exposures. So what we're trying to do with the exposome studies in the occupational, the wider environment, is to measure them, just measure lots of them, and try to evaluate patterns of exposure. Next one, please. Next one. Jon Snow. I presume all of you know this map, the Broad Street pump, somewhere in the middle, and he did a spot map. He just Jon Snow looked where the cases occurred and when they occurred, and identified the causes of cholera. So for many years, epidemiology worked with this type of approach, more elaborate than that, but basically that type of approach, having some kind of an exposure and trying to see where, with a spot map type, more elaborate, obviously, what happened. Next one. And we now have new tools. We have smartphones, we have satellites, we have data science capacity to look at interconnections, we have maps. Next one. And we have also from the other side, from the molecular side, we have the possibility of measuring in a very efficient way with small amounts of samples and in hundreds or thousands of samples, biomarkers that are related to exposure. On your right-hand side, you have the results of a urine sample of people of a study of swimmers. The orange is before you swam, the blue is after you swam. So by getting just a few drops of urine before and after, I can see what happens in your metabolome, basically. Very clearly differentiated. I mean, these are the ones that were clearly differentiated, but there are really lots of chemicals. On your left-hand side is just an indication of that we are working with much better knowledge of pathways. So you can evaluate exposures. It can be phthalates or dioxins or whatever, and we know much more the pathways. And if you're interested on how dioxins affect birth weight among weight of workers exposed to dioxins, you have ways of measuring different markers during the pathway. So you have capacity of interpretation of the biomarkers. Next one, please. And this we can do in high-income countries, and this we can do much of this in low-income countries. We have sensors and capacity to do things also in low-income settings that are more difficult. This is a study by my colleague here at ISGlobal, Catherine Tone, this is an air pollution, and she was doing this study in India, in a low-income part of India, rural India, and that is an example of one of the participants, and she would carry different monitors, different sensors, and you can see a camera also taking pictures, and this has passed obviously ethics committee, and the participants have the capacity to stop taking the pictures. And so we associate what people are doing using pictures from the camera with the information we get from sensors. Next one. And we have GPS, which is pretty standard in a way, and we can use cheap mobile phones that we give to many of our participants, and we know where the participants go. And we can evaluate also through mapping their exposures. And we can do that in any type of setting nowadays. Next one. Obviously, a big part of the exposome is not only the environmental exposure assessment, it's what happens in the biology side. So we, before we had the capacity of doing just genetics, doing SNPs and looking at the normal variation, of the DNA. And now we have the capacity of doing big studies on different omics. So, for example, how the DNA is transcribed, and we can measure the mRNA, and then we can measure how the RNA is translated into proteins, and we can measure proteins with proteomics. Next slide, please. Just an example, this is one of the studies. This is environmental, it's not occupational. There was an occupational component on people working in the swimming pools, but also just the results from the environmental part. What happens to people who went to swim in a pool? Pools have lots of chemicals. I mean, they're great, the pools, to do lots of things for back pain and having a good time, but they also have lots of chemicals. So this was a before and after comparison of people swimming for 40 minutes, and we measured things before and biomarkers, internal dose and biomarkers of effect after. Next slide. So just to give an example, this is what you saw before, metabolomics. We just measured before and after, and we could identify new pathways that we didn't know, basically, for exposure to disinfection byproducts that are prevalent in the pool. The next slide. And we could look at changes in the transcriptomics using mRNA and microRNAs. This is too complicated to explain, so I'm not going to explain it. Just to, it indicates that you have the capacity to look at hundreds or thousands, not hundreds actually, many thousands of changes, and tens in terms of microRNA, identify new pathways for the disease among people exposed and non-exposed. And you have to do special type of analysis to interconnect these changes. And the next one, or you can evaluate markers of effect. This was, we did it in macronuclei. And again, this is something we measure when the cell divides. It is quite laborious. And what you see in the top cell is part of the nucleus that has kept out of the dividing cell nucleus. So, which is a gross effect. So, and this means, something really gross has happened there. And we have been using it a lot in the occupational environment with buccal samples, urine samples, whatever, blood samples or whatever. Next one. And we showed that there was a lot of macronuclei among people exposed. Just to finish, then, this is one of the Exposome, the big Exposome project on occupational research effort, and we are, and has all sort of, I'm not going to go into details, all sort of different aspects. It tries to construct what Michelle was saying before, a mega cohort where we'll basically concentrate on, the environmental side, the occupational side, just have super big cohort, combining cohorts from different countries, and look at multiple exposures, multiple occupations with disease. And then we have specific studies, and I'm not going to go into details on that, on respiratory outcomes, asthma and lung function, and on night shift work, where that is based more on sort of classical exposome, where we are measuring a number of environmental exposures, and then we are measuring a lot of biomarkers and precursors of disease. So we will look at lots of precursors of cardiovascular disease. We'll do epigenomics, genetics, lots of pathways, on sex steroid hormone pathways, immune pathways, and try to make sense out of this much more complex picture. So I think I will stop here, and there are just a couple more slides, but it's let's stop here and see whether there are any comments.